Very good morning, dear student. Today I will start the topic nutrition in plants. It would be the second part of class 7th and subject is biology. Again, before starting the second part of nutrition in plants, I would like to go for the previous lecture. And in the first lecture, as you know that I have covered in the first lecture the photosynthesis process, and you can easily recall your first lecture, first topic, that in the photosynthesis, as you know that plants are prepared their food in the presence of sunlight with the help of chlorophyll and carbon dioxide and water. And in the result, in the photosynthesis process, you got the glucose, carbohydrate is prepared, and very important gas, oxygen is released out. So in the photosynthesis, you got the information that how does food is prepared. Why the green plants are autotrophs? Because they can prepare their food, and green plants contain chlorophyll. All of you have noticed that the leaf contain a green pigment that is called chlorophyll. All of you have knowledge that why all green plants are called autotrophs. In the second lecture, before the nutrition in plants part 2, I would like to tell you some important thing, that in the photosynthesis the chlorophyll is present in the leaf, and in the leaf upper epidermis and lower epidermis are there. When we see the leaf, you can see in the leaf, the structure of leaf, when we are going to start the structure of leaf, you will see in the leaf there are two surfaces, upper surface and lower surface. I am not going to tell you that apex, blade, margin. No. There are two surfaces. One is smooth upper side which is facing towards the sunlight, and the lower surface is little bit rough surface. So on these two surfaces the number of stomata are present. In the lower surface, in the dicotyledons, more stomata are present, and upper surface comparatively lesser amount of stomata are present. Today I will tell you the structure of leaf, and I will tell you the structure of stomata. So in this module, in this second lecture, the topic would be the structure of leaf and structure of stomata. So before beginning the structure of leaf and structure of stomata, I would like to tell you that the leaf, it has two surface, upper surface and lower surface, and stomata is made up of two guard cell, and the stomata helps in the exchange of O2 and CO2. This stomatal pore, stoma, which is called stoma, opens during the daytime when photosynthesis takes place, and it closes during night time when photosynthesis does not occur. In open stomata, oxygen is given out, released out, and carbon dioxide is taken in. So when we go for any structure, any topic, we must understand that what is the importance, what are the structure, what is the functions, what are the role of stomata. It's very, very important. as far as the structure of leaf, in the structure of leaf, you will notice that what are the upper epidermis, what are the upper layer, upper wall, what are the lower epidermis, what are the lower level, and where does these stomata are present. So in today's lecture, I will cover the stomata, I will cover the structure of leaf, and it's a very important. In these two topics you can prepare many questions for your unit test 1. That is why I am going to teach you the structure of leaf and structure of stomata. In the photosynthesis, both are playing very important role. In the photosynthesis process, as you know, in my first lecture I told you that how does photosynthesis takes place, what are the product, what raw materials are used in the photosynthesis process. So it is both the topic, both the leaf and the stomata are present, are very very important for the lecture. Coming to the structure of leaf. Before starting structure of leaf, I will draw the structure and you have to follow. You take your own time, ready with me, ready with your pencil, and draw the structure. And one thing is very very important, that you have to label at right hand side the symmetrical diagrams to create interest, to generate interest. You have to see how to draw the diagram, and you will also remember the structure of leaf. First you draw the outline so that the diagram looks nice. This is upper epidermis. One layer is prepared. And the same structure you can see here. Look at the structure. These are dumbbell shaped, and below the upper epidermis I am drawing, these are the now, these are the mesophils. Spherical structure are present. What you have to do for this structure, that upper epidermis you will label. There are 4 labelings. And mesophils. Below upper epidermis, these all the cells are kept in one category, that is mesophil. Look at this, I am just indicating for you. Look at this, mesophils, which contains the chloroplast, which contains the green pigment where the chlorophyll is present. You have to notice that in the mesophils, chlorophylls are present, chloroplasts are present. When there will be doubt in your mind, sir, where is chlorophyll? So it is present in the mesophils, which contains chloroplast, which contains the chlorophyll, which absorb the sunlight, and because of this chlorophyll, that photosynthesis takes place. You will surprised to know that non-green plants cannot prepare their food. They are not autotrophs, they are heterotrophs. Because of the absence of chlorophyll, non-green plants cannot prepare their food, they cannot perform the photosynthesis. So it is not necessary that all plants can prepare the food, all plants can perform the photosynthesis. No, non-green plants, it means all fungi cannot prepare. You have to note it down that non-green plants, non-chlorophyllous plants cannot prepare their food. They are not kept in the autotrophs. They cannot perform the photosynthesis process. In the photosynthesis process, the chlorophyll is very, very mandatory. The carbon dioxide is very, very important. The water molecules are very, very important. And sunlight is also very important. There are 4 raw materials. And we will be getting the glucose and oxygen will be released out. That is the next part of the photosynthesis. But before reaching to that next part, the structure of stomata and how does this oxygen is released, let us again come to the diagram. Now, as I told you, the four labelings. Number 1, upper epidermis. Epidermis means skin, surface. And here, look at this. This structure is lower epidermis. And the important thing, stomatal aperture, pore, very minute pores are there in the stomata, and I will show you in the practical experiment that where you can, under the microscope, you can see the stomata, what are the structure of stomata, how does it open, how does it close. It's very, very important. It's very interesting also in the biology when we go for the experiment, when we go for the activity, when we see these organelles structure, it would be very interesting for my students. So the next, stomata, stomatal aperture, you can see the small minute pore. So in this lecture, as far as the structure of leaf is concerned, you can see, you can write transverse section. When we dissect this leaf, you can also somewhere TS of leaf. Transverse, T stand for transverse, this longitudinal, the transverse section, internal structure I am telling you in this. So transverse section of leaf, it does contain upper epidermis, at the lower side lower epidermis, and number of stomata are more in the lower epidermis, you must remember. And this stomata is responsible for exchange of O2 and CO2. And between the upper epidermis and lower epidermis, all these things you can see, entire tissues, mesophils are called these, which contains the spongy tissue, which contains the palisade. It is for the higher studies for 9th and 10th class, but in class 7 it is sufficient for you. Now, so in the structure of leaf, you must remember upper epidermis, you must remember lower epidermis, and you must remember the mesophils, which contains the chlorophyll, which contains the chloroplast, which is absorbing the sunlight. It is not important that you have to draw the structure of chloroplast, but you have to remember the plastids are 3 types: chloroplast, leucoplast and chromoplast. And in the 7th standard, in the grade 7, you have to remember where the chlorophylls are present in the leaf. They are present, and what is the role of this chlorophyll? They absorb sunlight, and after absorption of sunlight, the carbohydrate, it means glucose, is formed. So in these structure you have to remember labeling number 1, number 2, 3 and the stomata. In the connection of this structure, now after this I will take you towards the structure of stomata. The number 2 is structure of stomata. I can erase this. Now, coming to the next, structure of stomata. As you know that stomata is minute pore present in the upper surface and lower surface of the leaf. So, before starting the lecture related with the stomata, it means what is the importance of stomata, what is the function of stomata. Let us see the internal structure so that you can remember for your future knowledge that what are the inner structure, internal structure of stomata. And by the diagram, when there are 3 H's involved, head, hand and heart, it means you are going to confirm your knowledge by involving writing work, memory power and heart. When there are 3 H, head, hand and heart, you can never forget, you can't forget these very matter for your whole life. So involve the diagram in the biology study. So coming to the structure of stomata, now you see and you follow me. Look at again. See, when I am drawing the diagram, you have to see the structure. These kidney shape structure, you can see, these are called bean-shaped, kidney-shaped. What are these? Guard cell. When I am drawing, look at this. When we are joining, you can see the stomatal pore is there. You can write here I'll show you this pore is open. When this is open stomata. I can also draw close. Look at. This is not in your course. For your complete knowledge, I am going to tell you that this is open stomata and this is Can you see it? Closed stomata. In the structure, you can move the stomatal pore is open, and the two kidney shaped, bean shaped guard cells are there, which turgid, which swells, and which is responsible for closing and opening of stomata. I will also explain in brief knowledge how does stomata open during daytime, how does stomata closes during night time, and what happen when the stomata are closed. Will there be exchange of O2 and CO2? Will the O2 will be released? Will the CO2 will be taken? No. When there is closed stomata, no exchange of gases will take place during night time. During daytime, stomata will be open because of the photosynthesis. When we take food in our stomach, it will swell, and by the stretch in the guard cell, it opens during daytime, and during night time, when there will be no photosynthesis, no exchange of O2 and CO2 will be taking place, and the stomata will be closed during night time. So vice versa, during daytime it opens. Why it opens? Because of the photosynthesis. Why stomata are closed during night time? Because of no process of photosynthesis, stomata are closed. Again, I am coming to the structure. These guard cell, you can label two more labelings. These are called chloroplast. In class 7th, there are two structures are very, very important. Number one is stomata, number two is structure of leaf. And in today's lecture, I am going to cover these structure of leaf and these structure of stomata. Chloroplast, another labeling is nucleus. So again, there are four labelings. Like in the leaf, there are four labelings. In the stomata, you can see chloroplast, nucleus, NUCLEUS. You can see, look at these. And the pore, the aperture is called stomatal pore or stoma. You should not confuse that is stroma, or it is STOMA. The spelling should be correct. And the lower one is guard cell. You can see these are the guard cell. So, in this lecture, the structure of stomata you have to understand. Now, coming to the next, that what is the importance of stomata? What is the role of stomata? What is the function of stomata? Now, only one function you have to remember. Stomata helps in the exchange of O2 and CO2. Okay? It helps in the exchange of O2 and CO2. As I told you that during daytime, when photosynthesis takes place and the stomatal pore will be open, look at this. Which gas is coming? Which gas is taken in the photosynthesis? You have already noticed in the first lecture that in the photosynthesis process, CO2 is taken in. And which gas is released out? O2. In the photosynthesis process, which gas is given out, which gas is released out. We are getting very important gas for our human life, for our respiration process. Which gas we take in the respiration? In the respiration, we take in O2. And here, in the photosynthesis, plants take in CO2. So, photosynthesis is opposite to the respiration process. For learning the equation, you have to remember that stomata, when these pores are open, which gas is entering? Look at this. CO2 is taken in, which gas is given out? O2. It means our plants are playing very vital role. They are not only producing the glucose, producing the carbohydrate, but they are also playing important role making our environment free of pollution. They are our friend. These plants are only the absorber of CO2. They stop the global warming problem by absorbing CO2. If there will be high percentage of CO2, the global warming problem can be there. So plants are playing vital role, performing the photosynthesis. Also they are absorbing harmful gas of the environment, and the CO2 is absorbed and oxygen is released out. So for your complete knowledge, I am going to give you 5 questions at the end of my lecture, and now you can write down the 4-5 questions. You can complete in your rough note, in your fair notebook of biology, the nutrition in plants part 2. For the recapitulation, I am again going to tell you the structure of leaf, structure of stomata, and in the structure what are the importance of these cell organelles, these structure, and what is the importance of stomata. So in today's lecture, the structure of leaf, you have to understand what are the chlorophyll, where does they are located, what are the stomata, where does they located, what are the inner structure of leaf, stomata, upper epidermis, lower epidermis, and mesophyll. And as far as the stomata or stomatal pore is concerned, in the second lecture, the stomata is responsible for exchange of O2 and exchange of CO2. Okay, so moving to the next, the assignment in the question answer. The first is draw the structure of leaf and stomata. Today's responsibility is related with the diagram, the structure of leaf and stomata. The label diagram. Got my point? There are 4 labelings in the stomata and there are 4 labelings in the leaf. Number 2. What are the chlorophyll? Where does they are present? Where they are located? Where they are found? Where they are located? Where are they located? Number 3. Write the role of stomata. Write the function of stomata. And the last question, you can again go for the important topic of this nutrition in plants. What are the role of guard cell? What is role of So role of guard cell, it helps in the opening and closing of stomata, because these guard cells swell during daytime after the photosynthesis process, and they prepare the food. They are in the preparation of food, they prepare the food. When the food is deposited, they swell, and by swelling, by turgidity, the stomatal pore will be open, and opposite to this, during night time, no photosynthesis process takes place, no glucose formation will take place, and the guard cell will not turgid, guard cell will shrink, and when they are shrinked, there will not be opening process, and the stomata will be closed during night time. That's it. That's it for the today's lecture, and hope we'll be meeting again with my next lecture. Thank you.